So last class we were looking at one of those applications. Applications in tissue culture, the protoplast. So the fusion of protoplast and how to get cytoplasmic hybrids. Hybrids. So that can also lead to having a haploid production. So for specific uh, work or specific aim, we need to select specific explants. Right, for wild hybridization or wide hybridization. So protoplast would be one of those selectable, the most desired explant of what we can take. So the next application is that how genetic variations can be obtained. So it's not introduced, it can be obtained using tissue culture, right? Using somoclonal variants. So a few of those varieties, especially in sugarcane, what is cultivated now. So that was obtained through somoclonal variation. So somoclonal variation. So the definition. So the soma is for the somatic cells. Soma means somatic cells. So the clones are those generations, like true to type generations that are obtained from the somatic cells, right? And the variation changes. The variation that happens in this vegetative cells that can be transmitted to the next generation. So these are all those somoclonal variation or those plants are called as somoclones, right? So the variation and somoclones. So these are all those same plants just with small genetic variation that is introduced in tissue culture. Right? And that should be of some positive nature, positive or even negative, that should be of some use. Right. It's like, for example, we'll see one example. So the somoclonal variation. So this entire topic can be in a 5 mark questions. What are somoclones and how to obtain the somoclones? Right. Somoclone, the first coined by Larkin and Scowcroft. So somoclone, according to them, the definition that's given as somoclonal variation is the genetic variability which is generated during tissue culture or the plant variants derived from any form of a cell or a tissue culture is called as somoclonal variation. So the phenomenon of high variability in individuals from plant cell cultures or adventitious shoots are called as somoclonal variation, or the plants will be called as somoclonal variants. So the variation in plants that have been produced in plant tissue culture. So when you get into this, so we go in for tissue culture to produce true to type plants, clonal plants, so which are genetically stable, which should be genetically stable. But sometimes, so due to that's extreme conditions in the sense like when we are subculturing those cultures. So if you are introducing some stress conditions, say like you are not cooling the forceps and then touching the explants with the hot forceps, then you are introducing like heat stress to those plants. So many of them will die off because of the heat, but some plants will make up or change their genetic makeup so that they can tolerate that stress, right? So that will be some genetic changes, and those progenies from those cells will have tolerance to that heat stress, right? Similarly, so if your media contains more of salts, NaCl, then many of those plants. So this is selection, right? How we can select or introduce those characters. So if we want to select for salt tolerance, so grow those cultures in a media containing higher amount of salts, NaCl, right? So many of those plant cells will die off, but those cells that can change or mutate and tolerate that salt will start to grow and multiply. So those will be the somoclones that introduces natural mutation to tolerate that salt, right? So this will be those variation that can be introduced in tissue culture. So these variations. So normally in normal times, there can also be somatic variation if we go in for many number of repeated subculture. So we say that the tissue culture can be maintained for more than 5 or 10 years. But that repeated subculturing will introduce some sort of stress in the plants, and those plants will change in the genetic making, right? So that will introduce some somatic changes, which may be positive or negative for us. So if it is positive, then we can take it, or if it is negative, then we need to throw those cultures, right? So such type of repeated subculturing will introduce this mutation and somoclonal variation. So the genetic mutation in plants that have been produced by plant tissue culture and can be detected as genetic or phenotypic traits caused by the in vitro conditions. Right. The genetic variation or inherited by the clones of the treated plant. So this should be whatever the changes, genetic changes needs to be like transmitted to the next generation, right? So we will see that there are two types. One will be transmissible and other is non-transmissible, right? The somoclonal variation may lead to desirable characters like increased pest resistance. So this is performed to achieve some beneficial properties in the plants like disease resistance or fruit quality, stress resistance, nutritional quality improvement, yield improvement, or any of those characters. We can breed or culture the cells in those stress in this stress condition and get a desired plant for any of those characters. What we are looking for. So this is one example in sugarcane tissue culture. So this is a natural. So this the popular variety of sugarcane that is still being grown, that is drought resistant and also with high sucrose recovery is this core 94012. Right. So this core 94012 sugarcane. So this was obtained through this tissue culture somoclonal variation. Right. So this type of variation can arise because of those stress conditions that is normally applied, that intentionally we apply, or normally when we go in for tissue culture. So when it is desirable, then we keep it as a variety. If it is not desirable, then just throw it. Right. So the repeated culturing, repeated subculturing will lead to such type of somoclonal variation. So the recovery of this genetic changes in the generated plants from somatic cells. So this gives natural variability. And to this variation of development of new varieties and for many number of characters. Through establishment of differentiated cell culture and defined conditions and the proliferation of number of generation and subsequent regeneration will lead to some characters that are good that we can keep it and transmit to the next generation. So such type of somoclonal variation. The one another one is soma is from the vegetative cells. So these the other types can also be from gametic cells, right? From the pollen grains or in the ovules. So if it arises such type of variation that arises in the gametes, that is called as gametoclonal variation, right? The somatic tissue derived variates from the gametophytic origin, such as pollen or egg cell, then it is called as gametophytic variation. Right? So this introduced variation among plants that is regenerated from the cultured gametic cells or called as gametophytic variation. So how it arises, the basis of somoclonal variation. So it occurs as a result of genetic heterogeneity in plant tissue culture. And that changes can get passed on to the next generation to the other. So this may be due to expression of this chromosomal mosaicism or also called as genetic disorders. So that can be changes in the genetic makeup, that can be SNPs or even large scale chromosomal exchanges, deletion or insertion can cause this somoclonal variation or spontaneous mutation due to culture conditions or physiological conditions. So this can also cause somoclonal variation. So the general features, the variation in the karyotype. Karyotype is the DNA content, the isozyme characteristics, morphology, the number and the structure of chromosomes. So this can be readily observed in this somoclones. So these are all the selectable properties, how we can select it like either in the media or just looking at the chromosome structures, we can select such type of chromosomes and this regenerated plants with the altered chromosomal changes. So they show changes in leaf shape, color, growth rate and habit, sexual fertility. So these are all those characters from which we can select those somoclones. So these are all these should be heritable mutations for to be used as a variety, right? If it is not heritable, then we cannot use it for further as a variety. So these should be the heritable mutations, and that should persist in the plant population even after planting into the field. And with these variations that can be both the qualitative and also quantitative traits. So for any of those traits, so we can select such type of somoclonal variation. So the types of this variation that can occur is the one is the heritable and the non-heritable variation. The heritable variation or those genetic variations that occurs in the DNA that can be transmitted to the offsprings. So this pre-existing variation in the somatic cells that can be transmitted to the offsprings, and these can be caused by mutations and other DNA changes, and these occur at high frequency. So which may be desirable or non-desirable variations. So other one is non-heritable variations, which is epigenetic. Right? Just DNA modifications that is not transmitted to the next generations. So the variation generated during tissue culture caused by temporary phenotypic changes, which may be due to culture conditions, varying nutrients. Say like, for example, so the repeated subculturing or too much moisture in the culture. So that will cause glassification of the leaves, right? Glassification of the leaves. So those are all temporary and that will not be transmitted to the next generation. So those are all epigenetic changes just for one generation. So this can arise due to the culture conditions, extreme culture conditions or varied nutrients. So this can cause such type of non-heritable changes in the plants, and these occur at low frequencies. So the variation that occurs in different types of cells. So in almost all types of explants, we can get this variation. Gametoclonal variation that is observed from gametic cultures. Clones of the gametes or protoclonal variation when it arises from protoplast culture, or called as protoclonal variation in from protoplast culture from the clones of the protoplast, or a caliclonal variation that occurs from the plants from the calyx, right? Or a mericlonal variation that can arise even from the plants that is obtained from the plant meristems, right? For any type of explant, we can get such type of somoclonal variation. So the causes of this variation, the physiological, genetic, and biochemical causes. So physiological cause. So the exposure of culture plants to growth regulators, like too much of growth regulators or extreme growth regulators, say like 240. So these are all extreme growth regulators. So they will be active only in very small concentrations. So if you add too much of it, then that can cause the heritable mutations, those plants. So that can cause somoclonal variation on the culture media conditions. So having more of sodium, sodium, so that can increase the sodium potassium channel, and this will cause this drought stress or salinity stress. So that can also give rise to the somoclonal variation. The genetic causes. So this is the important cause that will be transmitted. The culture conditions that may not be transmitted, that may be epigenetic. But the important one that is being transmitted to the next generation is the genetic causes. So there can be changes in even the chromosome numbers, aneuploidy, polyploidy, or monoploidy. So the gain or loss of set of chromosomes in aneuploidy or polyploidy. So that can cause increase in number of chromosomes. Monoploidy, so to get so even elimination of one set of chromosomes. So like what we saw in this bulbosum. So depending upon the culture conditions and the process, one set of chromosomes can entirely be lost, right? Which can cause to lead to monoploidy conditions, or the changes in chromosome structure, deletion, inversion, duplication, translocation. So anything, just like this X-rays, the mutagenic agents, all this changes in the chromosomes can occur in tissue culture, like deletion, even a segment. Segment is deleted, segment of chromosome inversion, a segment of chromosome is reversed or inverted, or duplication, addition of the chromosomes. Translocation between the arms of chromosomes. So any type of change can occur in this cultures, or gene mutations, or small changes, transition, transversion, insertion, deletion. So this change in chromosome structure involves a large portion of the chromosomes. So this gene mutation in a small section of the gene, right? So most desirable will be this gene mutation, not an entire change in the chromosomal structure. So this will be those most desired mutation that can happen, or that is advantageous to us in tissue culture. So the genetic cause, the aneuploidy or gain of loss of chromosomes. So from the parent cell, when you have six number of chromosomes, so there can be some meiotic errors during the culture, so that there can be unreduced gametes. So during this meiosis, it should lead to like 2n is 6, 1n should be 3. Right. So without any reduction in this during meiosis, this entire set of chromosomes is transmitted, so that you get the double or the tetraploid number of chromosomes. So this can be lead to the polyploidy or aneuploidy, right? The more number of chromosomes that can occur because of those changes, the errors that is caused during this division of these gametic cells, or the inversions. A chromosome segment, the entire chromosome segment is lost, or the chromosomes or translocated. The translocation, the entire segment of a chromosome is transmitted or translocated to another chromosome, or duplication, it's being repeated onto the another chromosomes, or inversion, so that can get inverted within the chromosome itself. So all this type of chromosomal changes you see in most of this X-ray or UV based like mutation, right? So that can also happen during this tissue cultures, or this plasma gene mutation, or cytoplasmic gene mutations. Cytoplasmic genes. So where do you have the cytoplasmic genes? In the organelles, right? In the mitochondria or chloroplasts. So such type of mutation can occur inside the mitochondria or the chloroplast. So this shows cytoplasmic inheritance. So such type of mutations. So this will be another most desirable, so that it comes only from the mother. So most this mitochondria chloroplast that gets transmitted only maternally. So this type of characters will be much more useful to develop a variety, or it can be because of those inversion translocation, may also be because of this transmosible element activation, or jumping GIFs that can also cause mutation via replication, recombination, or repair. So this can also cause inversion or duplication, and DNA sequence changes, direction, and altered fragment size using RFLPs, or change in protein, loss or gain in protein band, alteration in the level of specific protein, or DNA methylation inactivation, the transcription process. So this loss or activity, gain of activity of any of those specific genes because of methylation of DNA. All this can also again occur during this tissue cultures. So other cause or the biochemical causes. So biochemical cause that can happen as the lack of photosynthetic ability due to alteration in the carbon metabolism. So this is the most most seen variation that can occur in tissue culture. So even in our culture, we are seen in the tissue culture labs. So where we grow this rice explants or sugar cane, you see more of this albinos. Albinos is loss of chloroplast. So mostly this when Rubisco, Rubisco is mutated. Rubisco is one of those largest protein in the plants. So when that gets mutated, then that gives rise to the albino implants. So one of those most often seen somoclonal variation in plants is the production of albinos in plants. So there can be like deficiency of biosynthesis of starch, like carotenoid or nitrogen metabolism, or this can also give rise to herbicide resistance or antibiotic resistance. Right? Some characters, some of those characters that is like antibiotic resistance or herbicide resistance that are desirable to us that we can take up, but not these type of mutations, right? Which can cause albinos that is not desirable. So the process, you going for callus tissue culture. So going for organogenesis and then regenerate a plant, hardening and selfing. Then the screening during the hardening and the selfing of those plants, you regenerate the somoclonal variants. You select the variants, right? How do we select those variants? Right? And, okay, the factors affecting. So how this can be cost how this hormonal variation can be caused is using this selection agents. So we can that can be naturally induced. So this type of variation can be naturally induced, naturally happening, or this can also be induced by applying external agents, like external selection agents, like a toxin. So if we want to select for fungal disease resistance, add some fungal extract to the media, right? So those will be the fungal toxins. So only those callus culture that are resistant to this fungal toxin will be able to grow, right? So select those cultures. So select those plantlets that are resistant to those toxins or herbicide resistance, or add a little bit, a non-lethal dose of herbicide in the culture, so that only those plants that are tolerant to this herbicide will grow, and it will be naturally tolerant or mutated for that herbicide or amino acid change analogs for nutritional selection. So any of those selection agents can be added in the media for selecting for specific type of changes what we need, or selection of the propagutes, from which propagutes what we can get the cells, protoplasts, or the calli, and the duration of cell culture can also affect how we get the variation or how much we get the variation. The longer the duration of subculturing, the more the variation can be. And the growth hormone effects, just like we saw, more amount of hormone or difficult hormones like 240 can also affect the variation. Then the technique used for selection, what type of technique we use for the selection, then the stability of the resistant substance. That should not, the substance, what we use for selection should not get degraded. It should be stable enough. And what type of, in my testing procedure in what to select the plantlets. And so what tests we do to select those plantlets that are genetically stable, and the genotype, and again the same genotype and the explant source, as also some say on how this changes occur and the ability for the regeneration of the plants. So we need to be able to culture, regenerate and then harden the plants. So only then we can get any varieties. So isolation of somoclonal variation. So one with in vitro selection. So where we apply all these toxins, all other selecting agents, so that we can get more number of somoclonal variants, or without any in vitro selection procedure, naturally occurring variations can also occur. So without in vitro selection, so just get an explant derived callus, and then get shoot regeneration from those callus. You get the plant and transfer to the field and screen for the desirable traits or any agronomical traits, right? Like what we see here. So there is no induction here. So there is not a specific character that is being introduced. Just normally, we get the callus, regenerate the plants, and then put it in the field, and then test for presence of any of those characters. So this is hit and miss. miss objective, right? So we cannot get the desired character what we need. So without in vitro technique, so you take this callus and cell growth for the various periods of the medium without no selective agents and induce to differentiate whole plants, and these plants are cultivated in the medium supplemented with the growth regulators, and you get the callus, and these do not contain any selective agents, any of those selective agents. And these are normally subcultured, and they are grown in pots, transferred to the field, and they are selected for some of our variants. So the limitation, disadvantages of using without no selecting pressure is that there is no specific approach for isolation of somoclonals. Right? We cannot get any desired variants from this approach without any selection pressure. So the appearance of desired traits is purely by chance. And it is also a time consuming procedure. So you need to plant thousands of plants. So what we are looking for. So if you need to want for a drought resistance, then we need to plant thousands of plants in the field and check each of those plant has any drought resistance, right? Mostly it will not be any drought resistant. There may be other characters. It may be salt resistant or insect resistant, but we will not be selecting for them. There is no targeted approach in this without in vitro selection. So this requires screening of thousands of plants, and it is time consuming, money consuming. So generation of the with in vitro selection. So this is a targeted approach where we can select, we can induce, we can introduce specific character what we are looking for. Right. So first, so this was first given by Chalef, plant through Larkin, and so regenerative plants having said, okay, so these are some of those references where they have used this in vitro selection process to get desirable agronomic trait. So first we go in for the normal calic production, and then the next one with the selective agent. So if we need to select for pathogenic resistance or fungal resistance, right? So if we introduce the fungus, then it will kill the entire organism, entire plants, right? Instead, we can go in for the culture filtrate, which contains all the toxins, right? First, grow the pathogenic organism, then get the culture filtrate, there's a toxin isolation, and then put it and you determine the half lethality, 50% lethality, and then add it to the medium, right? So only those cultures, you go in for several of those selection cycles. So only those callus that are tolerant to this toxin will be good, right? So you isolate only those toxin isolation of the calli that are tolerant to this toxins, and going for regeneration and in vivo testing. And again, it need not be planted in the field. So it can be in vivo tested using either the toxin or the entire pathogen, right? In the media itself. So you don't need to go in for the field itself. So you go in for self-pollination or vegetative propagation, and you get the clones from each plant and test for disease resistance in the field. So this will be a desirable agronomic trait which has that disease resistance character, right? So by this, we can also go in for disease resistance, or instead, if we add more of salt to the media, then we can get cultures that will be resistant to salt tolerant cultures. So for specific conditions, we can introduce those specific selecting agent and selector with the in vitro selection conditions. With in vitro selection, so you introduce the presence of toxic substance or the selective agent, not necessarily toxic substance. If you want to select for cold tolerance, right? So introduce more of cold, cold, and only those cultures that are able to tolerate this cold conditions will grow, and select only those, right? Into the media under environmental stress. So you are introducing this environmental stress conditions, either maybe cold or high temperature, right? So high temperature, so only those cultures that are resistant to high temperature will grow. So select those cultures. So you get the explants and exposed to the medium containing the inhibitors, toxins, antibiotic, herbicides, or amino acid analogs, and you go in for the several selection cycles. So immediately, even within the first cycle, you will not get these cultures, right? If repeatedly. So how do this bacteria get antibiotic resistance? Repeated exposure of bacteria to antibiotic will give rise to antibiotic resistance in bacteria. So similarly, I expose this cultures for repeated cycles and isolate the tolerant callus cultures and regenerate the plants that is resistant to the toxin that are selected and grown further by vegetative or self-pollination. And this can be subsequently, the generations can be analyzed for disease resistance or any of those characters that we are selecting. So the advantages of in vitro selection, the specific approach. So we have a specific approach for isolation of a desired trait. Right? So we introduce a specific trait for selection. So because that we are not randomly selecting for a character. So this is less time consuming procedure compared with the in vitro approach. So the detection. So after we get all this plans, how do we select for those changes? So analysis of morphological characters. So just by looking at the morphology, the qualitative characters like plant height, maturity, then flowering. So all these morphological characters, quantitative characters like yield of flower seeds, wax contents, all these characters can be analyzed for morphological characters or cytological studies if there is any changes in the chromosomes. Staining of the meristematic tissues like the root tip, leaf tip with falgoner and osteopermine. So this will give the number morphology of the chromosomes. So we can see if there is any changes, changes in the morphology of the genetic changes or detection in DNA contents, variation in detective contents, the cytophotometric detection. So those stained nucleus can be used to measure the DNA content, whether there is aneuploidy or polyploidy, whether the DNA content has increased. Or detection by electrophoresis, the concentration of the enzymes, proteins, if there is any changes in the enzymes or proteins. Isozymes, if there is any changes in isozymes, that can also be detected, like pigments, alkaloids, amino acids can be detected by this electrophoretic pattern or disease-resistant variation by responsible where the disease resistant can be used as a selecting agent, even on the plants, using this actual pathogen or just the toxin extracted from those pathogen or herbicide resistant variant grow those plantlets by the addition of herbicide. And the detection of environmental stress like water logging, drought resistance, silk alkali, or high temperature stress or mineral toxicities. So these are all those examples that has been done in tobacco, tomato, or in high temperature tolerance in pear, or mineral toxicity for aluminum toxicity in sorghum plant. So such type of the stress can be introduced and selected and selected applying those stress agents. So the avoidance of this somoclonal variation, avoid long-term cultures or repeated subculture. Right, maximum we can go in for 5 or 10 subculture, like not more than 1 or 2 years. So if it is more than 1 or 2 years, then lots of variations will be introduced in those cultures. So using auxiliary shoot induction systems where possible, instead of going for repeated like meristem cultures, going for auxiliary shoot induction systems, like propagating chimeras by other clonal systems, like increasing number of subculture increases the likelihood of somoclonal variation, so that the number of subcultures in the micropropagation protocol should be kept to a minimum. So regularly going for the mother culture preparation, not just the subculture. So regularly reinitiating clones from the new explants, which will reduce the variability over time, like using this 240, so which is a harsh herbicide and also a hormone that can easily introduce this variation in those plants. So avoiding 2,4-D in the culture medium. The applications is that the production of agronomically useful plantlets. That is not that we will not be able to get otherwise possible. So just like what we saw, the sugarcane variety that was obtained through the somoclonal variation that has 2 or 3 characters. One is drought tolerance, highest growth, recoverability, and also red rot resistance. So all these characters was introduced at a time in this variety through somoclonal variation without any of our interference in that culture. So this resistance to diseases, like introduction of disease to resistance to diseases. So it's difficult to identify the genes responsible to overcome this disease, but somoclonal variation can give rise to natural disease resistance or abiotic resistance, herbicide resistance. So all this can be applied through this somoclonal variation. Improved seed quality, especially the scented varieties of geranium, woody ornamentals, like the sugarcane, the most used example used in India is the sugarcane selection for higher yield and red rot disease resistance. Or potatoes, yield and disease resistance, then leaf variation and horticultural plants. All this can arise through somoclonal variations. These are all the applications. So these are all the examples that we can obtain. So for rice, a reduction of flowering period, panicle size, sterility mutants, soybean, then tomato, dwarf habit, and carrot, higher carotene content, tobacco, increased yield, type of inflorescence yield. So these are all those characters that has been selected specifically in many of those crop lines. So resistant to diseases. So this is one example. And one example, the resistant first reported in sugarcane for eye spot disease and Downy mildew disease and Fiji virus disease, regenerating plants from the callus of susceptible crowns and screening for the smoke wounds. And this is one specific example from sugarcane that has been developed and also being grown normally. So these are all those using like in vitro selection procedures and different varieties has been developed. Say like for example, rice, helminthosporium, leaf spot. Leaf spot, use the extract from helminthosporium and then select for those cultures. Rice, barley. So what they have used for rice, they use the crude toxin from the bacterial cells or maize, the tea toxin for the wheat, crude toxins, neomycin from helminthosporium. So all this crude filtrate, virus filtrate has been used for tomato mosaic virus. So such type of selecting agents can be used to select to get resistant varieties. Resistant to abiotic stresses. So you introduce the specific stress like a freezing tolerance in wheat or salt tolerance in rice, maize, tobacco, or heavy metal, aluminum tolerance in other plants, drought tolerance, herbicide resistant characters. So like in soybean, imethacillin resistance. So by selecting, these are all those examples that has already been up time, like for tobacco, glyphosate, carrot, glyphosate resistance, lotus resistant to 2,4-D. So all these plants has been already developed by using the specific like herbicides for selection. Also improved seed quality in lathyrus. In lathyrus, for reduction in the neurotoxin. Say like in the lathyrus is this masurda, right? Masurda, Masurda. So that has more of this neurotoxin. So by selecting for this, there is a reduction in this neurotoxin from this lathyrus. Letter is which is a variety bio L212 with reduced neurotoxin contents through somoclonal variation. This is another example for improved seed quality. So the advantages of and the disadvantages of somoclonal variation. So helps in crop improvement, additional genetic variability, more of secondary metabolites, toxicity is reduced. This can be used for and stable cell line can also be preserved for cryopreservation. And example of a citronella with more of oil. The disadvantages is that it requires extensive and extended field trials to see if this is stable, right? So more of this needs to be stable in the field. So first, so we introduce the toxin to the plants and develop a resistant plant. But so how do we introduce? So because of the presence of that herbicide or toxin, the plant develop resistance. And it can also be opposite. So when the toxin is removed, then the plant can revert back to its norm. So it is possible that because this is normally happening in the plant, so introduction of toxin will get resistant plants, and removal of that toxin then allow the plant to come revert back to its original condition. Right. So this requires extended field trials. So it should not revert back to its original condition. So only such type of plants we need to select, which requires extended field trials. So and also many of the times, not sometimes, many times leads to undesirable results, which reduce fertility, growth rate, and overall performance of the plant. And these are genetically unstable and non-predictable. And the tissue culture and complex agronomic traits not suitable for complex traits. Right? When it is controlled by an entire loci, it may not be suitable. Then it may also develop pleiotropic mutations. So even with the single, that can be many characters, many characters with the need with our character. Not just if we select for herbicide tolerance, there can also be disease susceptibility. Right? So there can be many pleiotropic effects that can arise due to this. Not it is not like pointed mutation. Like there can also be many changes in that. Plants often lose the power of regeneration. So these are all those disadvantages. And coming to this, the same with when we use this gametes, it will be the gametoclonal variation which give rise to the haploid plantlets, and this meiotic crossing over and that can happen in the gametic clonates, and this can be stabilized. Those characters that is introduced here can be stabilized by doubling the chromosome number, and we get a doubled haploid with the desired characters. The production of gametic clones, it is similar to whatever we get in the normal clones. Improvements have been made through this gametic clones in rice, wheat, tobacco. Gametoclones obtained in rice or plantain, almost all of those characters, what we saw for somoclonal variants. So the cell culture technique may introduce variations, and a variation may be introduced even while doubling the haploid chromosomes. So like when we add a colchicine, that will not necessarily double the chromosomes, right? So normally, so the doubling of the chromosomes and colchicine doubling of the chromosomes happens because of the disintegration of the microfibrils, right? So sometimes, if it is not enough, there can be some microfibrils which can give aneuploid structures, right? So which is not stable, doubling the haploid, right? So you will get an aneuploid, not a dihaploid. So genetic variation can also occur due to the heterozygosity of those dihaploids. Applications, gametoclone suitable for breeding, crop improvement, disease, herbicide, toxin resistance, abiotic resistance, biotic resistance, insect bacteria. All this can also be introduced using the gametoclonal variation.